Mitosis is the cell division in eukaryotes, and eukaryotes are those cells that have a nucleus, and you see over here that's supposed to be a nucleus. It is a nucleus. It's from a plant cell, and in this nucleus you have DNA, the so-called the chromatin DNA, and during mitosis, during cell division, the DNA has to be moved around in a cell so that every daughter cell has the same DNA. And that's essentially the whole purpose of mitosis. How can we move around the DNA in a cell so that both cells get the same DNA? And I started off my explanation with interphase, but it's important to understand that interphase is not part of mitosis. Interphase is the non-dividing time of a cell's life. So cell does not no division. Okay, it's the time between the cell division, but it's itself not part of it. But I have to mention it here because there are some important things that go on in interphase, namely the replication of DNA. The replication is it's a fancy way of saying the copying of DNA. We have to have two cells at the end after division, so we have to copy the DNA of the parent cell first. During interphase, of course, also proteins are made because that's basically what cells do when they live, and the cell also grows in size. And what you see over here is you see that the nucleus is visible. That's maybe also something that you would like to add over here. Nucleus visible. The nucleus is visible over here, and the thing that's so red in here, that's our DNA. And the thing that you see out here, that's the cell wall, because these happen to be plant cells. Yeah, it's also a cell wall over here. Okay, so you get the idea. And this is my nucleus, of course, and there is DNA in there. That's basically the cell. Which we've drawn when I talked about in a different video about cell structure. I've also drawn all the cell organelles in there, the nucleus and so on. So that's basically the cell as we know it. And I'm not going to simply, well, if you want me to, I can draw it again over here. So what I generally do is that's the cell membrane. And over here I have the nucleus visible with the DNA in here. Yeah, so that is my chromatin, which is the loose DNA inside the cell. So now let's move on. The cell has grown in size, and now the cell decides it's time to divide. And now the following thing happens. We go into prophase, and prophase is the first stage of cell division. And look over here in the picture, you see that something interesting happens. You see it's not as homogeneous anymore. It's kind of like it's a little bit granulated, and there's some white parts over here in between. And what happens right now is when you see something like this, is you see that the DNA starts to condense. The chromosomes form out of chromatin. So these things that you see over here, these are already chromosomes which start to form. All of these things over here, okay. These are already forming chromosomes. That is really important because the cell now needs these chromosomes because we have to move them around inside the cell. The nucleus starts to disappear. You can still see a little bit of a nucleus around here, but it's in the process of dissolving. The nucleus or the nuclear membranes disappear, and so-called spindle microtubules form. You cannot see this very well over here in the light micrograph because they are so fine. But essentially, what happens is that we have so very fine protein fibers which stretch through the cell and which also attach to the forming chromosomes, and that's the spindle. So they're very, very fine, and there are many of these fibers around. Okay, so and in a plant cell you do not have the centrioles over here. In an animal cell you have centrioles, so maybe I should not draw them because it's a plant cell. Okay, I'm going to take this away again. Okay, so I've also erased one of these spindle fibers. So that is basically the spindle microtubules. So, and I'm going to make a drawing right now of this so that you also have a nice drawing in your notebook. This is my cell. And the nucleus starts to disappear, so I'm going to draw a dotted line over here. And inside the nucleus, I'm starting to draw my chromosomes right now. These are my chromosomes. I know this is very schematic. It looks very different than over here, but simply for the sake of understanding, I should also draw a few smaller chromosomes. Yeah, and maybe one over here. Okay, so, and strictly speaking, I kind of aligned them already, but there's no reason. Maybe some chromosomes are, I don't know, yeah, on the side like this. Yeah, why not? It's a little bit, yeah, scattered throughout. And over here we have the spindle microtubules starting to stretch, and they also attach to the centromere of the chromosomes. Yeah. Up, you get the idea. Okay, so that is basically what happens in prophase. And a few more of these spindle microtubules. So, okay, that's basically it for this stage, and we move on now to the next stage of mitosis, which happens to be metaphase, and the chromosomes align at the equator, that is the middle of the cell. How do they get there? Well, the spindle fibers, the spindle microtubules pull them there, and over here in the picture you can see these are the chromosomes right now. That's a chromosome over here. You can a little bit barely, you can see that maybe it's made of two different parts down here, and so the two sister chromatids you might be barely able to see. Okay, so that's another one over here. Okay, over here is another one, and this one over here, that's the equator of the cell. Well, if this is the equator, then these, of course, here and here have to be the poles of the cell. So we use an analogy from geography here. If they align at the equator, you might wonder why is it kind of sticking? Why are they kind of sticking out like this? Because when the spindle fibers pull them to the equator, then the arms of the chromosomes bend backwards, and that's why it kind of sticks out. I'm going to make a drawing over here again. And I have my spindle microtubules stretching the cell. Now I'm not going to draw a nucleus, of course, because the nucleus does not exist anymore. And I'm going to draw now over here at the equator of the cell. I'm going to now align my chromosomes. Actually, I have to align very many of them. I'm just going to draw a few of them. You get the idea. That is the equator. And it does not look like this, of course, is because as we pull them to the center, the arms bend backwards. So over here, I'm going to draw it a little bit different. I'm running out of space. So if this is the equator over here, you can actually see that the chromosomes, as they're being pulled to the middle, the arms bend backwards, and this one here is the spindle microtubule, and the chromosome which is over here on the other side and pull over there, it will look like this, that the arms of the chromosomes pull backwards, and that's the spindle microtubule. So what you have at the end is you have a cell. I'm gonna make yet another drawing which looks a little bit like this. If the equator over here, yeah, then you have the arms of the chromosome kind of sticking out from the equator to the side. Okay, and that's basically what you have over here. But schematically, in a simplified way, you would actually draw the chromosomes aligned like this because it's more easily understandable. But under the microscope, it looks like this. You have the arms of the chromosome sticking out to the side because they bend backwards as the spindle fiber pulls them. What is the point of this? The point is we want to create a form of a symmetry, because the two daughter cells that form have to be the same, and therefore we have to make sure that the left side and the right side basically are the same. But if the chromosomes are scattered randomly throughout the cell, we do not have the symmetry. So we move on now to the next stage, and the next stage is anaphase. And in anaphase, the centromere splits and the sister chromatids separate. Let's have a look over here again. This is a cell, and you can see that that's a pole of the cell, and you can see that the chromosomes are kind of sticking out to the middle like this. These are all the chromosomes, and the same down here as well. You have chromosomes sticking out towards the center. And why does it look like that? It is because what happens is, and I'm going to draw it a little bit bigger right now. You have the sister chromatids. I'm going to draw it like this. And over here is spindle fiber. They separate, and they are now pulled to the poles of the cell. So the centromere, which held the sister chromatids together, now splits, and the sister chromatids, the identical DNA, is now separated. So that's at the very beginning, but as we pull it, remember again, the arms of the chromosomes bend backwards over here and also over here. And it would actually look a little bit like this. Bends backwards. Okay, and over here as well. It bends backwards. And as we continue pulling it, when it's almost at the end, we would have something like this. The spindle fiber is now pretty short, and the sister chromatids still bend backwards. And if you have many of these, then it looks like this over here. Now I'm a little bit out of space, so I'm going to draw it simply into the picture over here because I'm a little bit out of space. So that's my cell over here, and the spindle fibers are really short, so all the arms of the chromosomes kind of looks a little bit like this at the very end. You've got many of these chromosomes now. Okay, and yeah, that's basically what you see over here. And this basically simply explains the process and explains why it looks like this, that you have the things now pointing towards the middle. So that is basically the first time when we start to separate the identical DNA. But remember, we still only have one cell. So far, up to this point, we only sorted out the DNA within the cell. But the cell, strictly speaking, has not yet divided. It's an important part of the cell division process, of course. But the cytoplasm itself is still present as one unit. So now let's move on to the next stage. This happens to be the last stage, and is known as telophase. And telophase is easy to remember because it's a reversal of prophase. So the nucleus reappears, the DNA decondenses to form chromatin again, and you have a process called cytokinesis at the end of telophase. Now let's have a look over here at this picture over here on the right. These are of course interphase nuclei. Well, that's basically something that we do not deal with right now. This one over here is prophase, so it's cut off here. These two cells over here, this one over here and this one over here, we just talked about, that is anaphase. You see that the sister chromatids have separated and are pulled to the poles of the cell. So now where is telophase? And if you look over here, is that this cell over here is a telophase cell, and I'll explain right now. So first of all, you have the two nuclei reappearing. That's one nucleus and that's another one. We still over here have one cell, but you see these things here, these lines in the middle? That's the rest of the spindle fiber. Yeah. So it's still needed for making a cell wall in this case, but you can still see it, and it's still one cell. I'm going to erase this again. So you see that this one over here is a telophase cell. So when they ask you explain what telophase is do not just simply say it's a reversal of prophase, but you have to say it's the nucleus reappears, or the nuclear membranes reappear, the DNA decondenses, and so on. However, there's now an interesting new thing at the end. It's called cytokinesis. And cytokinesis is the physical splitting of the cytoplasm. So when there a new cell membrane and a new cell wall is made. But before I talk about cytokinesis, so that you again have a drawing in your notebook, I'm going to draw a cell over here. And over here, I'm drawing the reappearing nuclei, and in here you start to form the loose chromatin DNA again, and the spindle fibers also start to disappear, but you see that at least the beginning of telophase you only have one cell. So in the next foil, I'm going to explain cytokinesis, which is a part of telophase, and it's a really short part. And the cytokinesis is the division of the cytoplasm. It is different in plants and animals. And I'm going to now show you how cytokinesis works in plants, and how cytokinesis works in animal cells. In plants, what we have is the following. Is we have our telophase cell over here with the two nuclei reforming. And because we have a cell wall on the outside, which is pretty hard and rigid, the plant divides by forming a new cell wall in the middle over here. New cell wall, and of course cell membrane. So maybe I should draw the cell wall a little bit thicker over here, because the cell wall is fairly thick, and of course also here in the middle. You get the idea. In your notebook you can draw it a little bit nicer than what I'm doing right now. So you're starting to build a new cell wall from the middle, and the spindle fibers that we still saw in the picture, they are now responsible for bringing the substances to make the cell wall and the cell membrane to the middle of the cell. So there are little vesicles which are attached here, and now the spindle fibers are used to carry these vesicles with all the building materials to the equator of the cell. I'm just mentioning this, why you might wonder why the spindle fibers are still present, because they're now used to transport substances to make the new cell wall and cell membrane. And because new cell wall and cell membrane sounds kind of, well, not fancy enough, we call this new cell wall, we call it a cell plate. A cell plate. So plants have cell plate formation during cytokinesis. And that's necessary because the cell wall is very strong and not flexible. In animals, now let's have a look at the animals over here. Animal cells over here, the situation is a little bit different. We have also our telophase cell, but the middle of the membrane of the cell starts to fold inwards. Okay, see over here it kind of folds inwards, up, not very nice. And over here I have a nucleus again and a nucleus again. It's possible because there's no cell wall over here, and what do you think is going to happen? It starts to move towards the center until the membranes join up, and then you have two cells, and that's pretty much it. Yeah, so here you have an infolding of the cell membrane. Infolding, hope you can read my handwriting. Okay, infolding of the cell membrane to form two cells, and and that's basically it. So you see that is one of the differences between plant and animal cells, is that they have a different way of doing cytokinesis. So, and that's basically it for right now.